do have a disclosure to make. I am the inventor of the SatiSphere device, which I will speak on today, and I'm a consultant for the company EndoSphere. Well, I thought that obesity was a uniquely American problem, but then I saw this photo and I realized that it's actually going global. Well, how does this happen? Well, we don't really know. We know it's multifactorial in terms of causes, environmental and genetic. But all treatments are rooted in a perspective of what the cause of the disease is. And at least historically, the cause of obesity has been felt to be a problem of willpower. It's a matter of an individual's choice to become obese. It's not a disease. And therefore, the treatment follows that the individual must be forced to either eat less or to absorb less. And it's really against that background and foundation that what I will call the surgical paradigm was developed. To eat less, for example, with a sleeve gastrectomy where a large portion of the stomach is resected, or to absorb less, the classic jejunal ileal bypass, which of course was abandoned because of the devastating side effects of malnutrition, or a combination of the two to eat and absorb less, and that would be the gastric bypass or the duodenal switch procedures, which are very popular today. And endoscopists have modeled their treatments after this surgical paradigm. So of course the gastric balloon was the very first treatment to be introduced, and it occupies a large space in the stomach and forces the patient to terminate eating, and also at the cost of much nausea and vomiting. And we've developed various platforms for suturing and stapling that enable us to mimic, at least, what the surgeon does with a vertical gastroplasty. The lap band is a lesser invasive surgical approach, creating a 15 to 20 cc pouch. And now endoscopists are attempting to recreate the same as a lap band, but internally, of course. Here are examples. This is the restrictive pouch, the Terrace device, and this is an intussusception cuff called the trim. And so these try to recreate essentially the same effect as the lap band, a small gastric pouch. And endoscopists have even succeeded at creating platforms to force less absorption. And this is the endobarrier device, and it consists of a sleeve some 60 centimeters in length and extends down to the distal jejunum, and it is anchored in the duodenal bulb. And we even have a platform that mimics the gastric bypass or duodenal switch, to eat less and absorb less, and this similar to the sleeve that you just saw, but it's anchored actually at the cardia and extends down the length of the jejunum. This is called the endoluminal bypass. Well, whether it's done surgically or endoscopically, all of these modalities are based on the surgical paradigm, and this in turn is rooted in negative conditioning. Essentially, the patient will develop pain or discomfort or side effects when he or she eats, and this in turn will result in termination of eating and loss of weight. We know that the space restrictive strategies will fail over time, and after all, that's why the surgeon will create a gastric pouch in the gastric bypass procedure that is the size of an egg, anticipating that this will stretch over time. And patients find ways around negative conditioning by consuming high caloric slurries. The malabsorptive strategies bypass the duodenum, and of course this is a problem for the endoscopist because we no longer have access to the bile and pancreatic ducts. And there are many complications of surgical tissue remodeling and invasive implants that come to the endoscopist for treatment: leaks and infections, stenoses, obstructions. So in my view as an internist, obesity isn't a surgical disease, it's a metabolic disease. There's not a diseased organ or tissue, an inflamed gallbladder or appendix that needs to be removed or repaired. It's a disorder of energy homeostasis. There are physiologic pathways that regulate appetite and energy metabolism, and these are intact, but they're inadequate. So the body is not able to cope in a man-made environment where high caloric foods are overly abundant and our lifestyle is excessively sedentary. And understanding these pathways is key to developing smarter treatments. Now, we have accumulated a wealth of knowledge today on the neurohumoral regulation of appetite and energy metabolism. The gut-brain axis was described about 2 decades ago. We know today that there are various gut peptides or hormones that interface with various parts of the brain. We also know that there's a gut-gut axis where various organs of the digestive disease tract communicate with one another to regulate energy homeostasis. And there's even a fat-brain axis. The fat has been shown to be an endocrine organ in itself and communicates with the brain. There are a number of hormones that have been isolated that constitute the gut-brain axis, and we heard about one of these, ghrelin, in Dr. Kim's presentation. In the duodenum, we have cholecystokinin and gastric inhibitory peptide and incretin. And in the ileum, we have peptide YY and GLP-1. Cholecystokinin was described first about 3 decades ago. It is produced by the eye cells in the duodenum. And it is stimulated to release CCK both into the bloodstream but also as a neurotransmitter by fatty acids and proteins that enter into the duodenum. The CCK that enters into the bloodstream, into the portal vein, then crosses the blood-brain barrier and interfaces with the forebrain. And the CCK then that acts as a neurotransmitter activates the vagal afferents. And these vagal afferents in turn trigger or communicate with various centers in the hypothalamus and the hindbrain and the brainstem. And the result is that meal size is reduced, and we call that satiation. And the desire to eat between meals is suppressed, and we call that satiety. So even though we call these satiety hormones, really they are satiation hormones. This is a busy slide, but it illustrates the central role that CCK plays in the transition from the ingestive phase, where food enters into the stomach, to the digestive phase, where food now enters into the duodenum. This is where digestion starts. And CCK has a amplifying or stimulatory role for a number of organs, like the gallbladder to contract, or the liver to release bile acids, or the exocrine pancreas to release pancreatic juice, but also stimulates the beta cells of the pancreas to release insulin. So CCK in itself is actually an incretin. And it inhibits stomach motility. So it creates gastric paralysis. And it results in contraction of the pylorus, not shown here, preventing food from exiting the stomach into the duodenum because the body has now shifted from an ingestive to a digestive phase. CCK is the only confirmed physiological satiety peptide. There is a temporal link between nutrient-stimulated CCK secretion and the inhibition of food intake. This has been demonstrated in many studies. The infusion of CCK at physiological doses, not at pharmacological, but physiological doses, reduces food intake, and it stimulates the feeling of fullness in humans. And in animals, it reduces intake when the animal has an open gastric fistula, that is to say, a sham-fed animal. The CCKA receptor, called CCK1R, when we block this receptor, it antagonizes the satiating effects of CCK and it increases meal size. So a great deal of evidence supporting the role of CCK as a satiety peptide. We also know today that leptin, this is the hormone that was discovered at the Rockefeller Institute, produced by adipose tissue, that the leptin interfaces with CCK and it amplifies the action of CCK. So with this evidence, it is clear that the duodenum is a key organ in energy homeostasis. Now let's look at some of the evidence for this. Well, we have sham feeding experiments in rats, and this goes back to the 1970s. Now, these are rats that have open gastric fistulas. These rats will eat incessantly. They will not stop eating as long as the food is able to leave the stomach through the fistula. It was then shown in the mid-70s that when liquid food was infused into the duodenum in these sham-fed rats, the rats stopped eating. And we called this intestinal satiety. And it was also shown that when CCK was introduced intravenously, it inhibited feeding in the sham-fed rats. Another level of evidence is that when they looked at pyloric occlusion studies, so the animal is given a cuff around the pylorus to prevent emptying of the stomach, that the animals with cuffs open ate less than cuffs closed. Now you would think the opposite, would you not? So when food can enter into the duodenum, the animal actually terminates eating. And we have clinical studies. For example, here in a trial of 20 males, they were given either lipids, glucose, or saline infused directly into the duodenum. And they found that the lipids decreased intake by 22% compared with sodium chloride. And lipids decreased intake by 15% versus glucose. And we also have studies where glucose was infused intraduodenally. This reduced hunger and food intake. But when glucose was introduced intravenously, given intravenously, it had no effect on eating behavior. And because of the interest in time, I'm going to skip this, but just mention that now there's research being done on metabolites of fatty acids, the long-chain fatty acyl coenzyme A, and its function in suppressing glucose production, which is called the intestine-brain-liver axis. And this cartoon here summarizes the three pathways in the duodenum. In the red, we have the intestine-brain-mouth circuit, that's suppressing eating, mediated by CCK from the eye cells. In the purple, we have the intestine-brain-liver circuit, mediated by metabolites of fatty acid. In the green, we have the intestine-brain-pancreas circuit, mediated by GIP and incretin. So what we need is a smart device that, for example, can reduce flow in the duodenum. This will result in an increase of contact time of byproducts of nutrition with the lining of the duodenum, and it will distend the duodenum. And this will trigger the chemoreceptors, the baroreceptors, and the mechanoreceptors. And in addition, the device may have contact with the duodenal wall and stimulate the mechanoreceptors. And all of these, mediated through CCK and GIP and fatty acid metabolites, will induce satiation and satiety and may be insulinotropic, may have a beneficial effect on diabetes. And this is what such a device can look like. This is the SatiSphere device, and you can see a C configuration. It is self-anchoring in the duodenal C, so it has conformational stability, and it consists of these open mesh spheres that act as speed bumps. And this cartoon here will show how the device is implanted through an endoscope working channel, and it's delivered no different than how a stent is delivered. So you can see the scope being retracted as the device is advanced forward, and it is positioned to accommodate the C configuration of the duodenum. Now the food, the ingestate, encounters these speed bumps, and the contact time with the duodenum is increased with the duodenal mucosa, and also it distends the duodenum, triggering the baro and mechanoreceptors. And here you can see that the device is easily removed also through the endoscope. And this video will just very quickly show how it looks endoscopically. This is no different than how a stent is deployed. It takes literally a couple of minutes to deploy this device. There are no anchors, there are no barbs. This is completely non-invasive. It is a simple and smart device. Simple because it's easy to deploy, it's a quick procedure, no special skills are required, no special equipment. It's smart because it is based on a physiological mechanism of action. It preserves native anatomy. It is adjustable, it's removable, and it can be reimplantable. So in conclusion, the surgical paradigm, which is mechanistic, is gradually evolving, and we're going to hear more about this in the subsequent talks, to a physiological paradigm. And smart endoscopic treatments that amplify physiologic neurohormonal signals of satiation and satiety are needed. Thank you very much.